Okay, well, thank you very much to the organizers of the issues class, Jack and Charlie and Sue, for inviting me to do this and enabling me to be able to do this and kind of share part of my world with you folks. So the title here just tells you maybe a little bit about me. So a little bit about my background. So I have, and I guess I'll introduce myself. Everybody here knows me as Wally. I have training in proteins and what proteins are and how they function and what they do. And probably my most profound influence was a time that I spent at the NIH, and I learned an awful lot about proteins and how they function. Had an opportunity to make proteins, to clone genes, and really get a feeling for how they function. So I've been back local for quite a number of years. I've been in the chemistry department at Lebanon Valley College here since 1999. And I don't think I say anything here that's maybe too troubling or challenging or whatever, but always when I give a talk like this, I always share that my thoughts, opinions, and ideas here are my own. So I need to start off with, and I shared with Jack here a couple minutes before we started, that maybe the majority of this talk is not about COVID proteins, but there are some things with the proteins themselves, with proteins, that I think you really need to understand in order to get my perspective. Proteins themselves are really kind of simple when it comes down to it. And so proteins are composed of amino acids. There are 20 common amino acids here. There will not be a quiz at the end of this on any of what you see here. But I put this up here to really show you and drive home the simplicity, but how we actually get to the level of complexity that we do when we actually work with proteins and deal with proteins. You see here, everything that's not in color, that's here, all this stuff is the same for every one of these 20 common amino acids. Now, there are some other little things that will work their way into proteins here, but this is pretty much it. This is what makes a protein a protein. So we take amino acids and we link them together, and we find out when we look at proteins are in fact polymers, and you actually have amino acids that join together here. Here is the foundational structure of a protein, where we take amino acid number 1 and we link it to amino acid number 2, 3, 4, and this keeps on going and going and going. Now, simple. Proteins are composed of 20 amino acids, but now when you think about the possibilities, all the permutations that actually can exist for a given protein. So we have 4 amino acids in this simple structure. When proteins are really tiny, we call them peptides, but we have 4 amino acids here. Every one of those 4 positions could be one of any of those 20 amino acids. And there are some odd things that you can kind of throw in there, some special amino acids and things like that. You pretty much get your choice of 20. And so here, if you actually take and look at this kind of with a mathematical point of view, all your possibilities for what a protein could be, for every position that you add here, you add another number to this. So for this structure up above here, there are 20 to the 4th possibilities for what that protein could be. That means here that although everything starts off really simple, to say, wow, only 20 common amino acids that you can use to make a protein, turns out for this short little stubby thing here, there are 160,000 different possibilities for what that molecule actually could be, which is, to me, again, simple but complex. To me, that's amazing. That's amazing. So what's the important part of this? The important part of this polymer are these R group things here, the things that were in color up above, those little special things that actually take and provide a little bit of character or chemical flavor to a particular amino acid. But what's really important to us, and I think lots of people here probably remember enough high school chemistry, okay, or whenever you may have encountered it, to know we see a couple things here. We see oxygen, we see nitrogen, we see hydrogen. Those really become the important parts of this structure. And we're going to talk a little bit in a moment here why those things are really so important in a protein. So all of a sudden here, we're starting to build this model, this idea of what a protein actually is. A protein is nothing more than a string of amino acids. And now we've got some atoms here that are actually really important for who a protein is and who a protein will become. So when we consider here adding those other features, remember up to that first table there, adding those other features where you have those things that were colored there, those different functionalities, different atoms, all of a sudden you take what was here, kind of the core part of that molecule of a protein, which is that right there, which we actually wind up calling the backbone. So that actually is what we call the backbone of a protein. Now we hang all these other interesting and fun things off the side of it. Now we actually start to make these polymers that are very, very complex. So we start to take individual amino acids, we link them together, the complexity builds, and you can imagine that these things can get really, really big and really, really long. Now, I actually want to introduce something here that actually makes this even more complex, and I call this molecular gymnastics, okay? So hopefully we're okay with the point that we link amino acids together, we start to build these strings of things, but now we actually have the opportunity to take this and twist it. Just about every place that you see a line in here, we can take and we can twist and we can do all sorts of fun manipulations of this. So I like to think of kind of like an erector set where we take the pieces, and you know how you link the things together, you put the little screws and the little bolts in there and put the nuts on them and tweak them, and kind of how you can still kind of tweak it until you get it right. It's kind of what this is like. So we have here a system of molecules that actually leads to complexity, So remember I said that those hydrogens, oxygens, and nitrogens were really, really important. Well, they kind of like each other. They like to interact with each other. And what happens here is we have within this molecule, you've seen some of these things up above here, and my handwriting is awful. So, and you know, with all the amount of typing that I do here, I write less and less and less. So it usually takes me a little bit of time to warm up and get these things flowing properly. And just, we're about to work out the cobwebs from the previous day. But it turns out that these atoms within a protein, they like each other. They like to interact with each other. And so we take that string of amino acids from up above that we've put together, we've actually made these polymers. And now you see that start to happen, okay? Probably everybody here in the room would recognize that and maybe even have a term for that. I tell my students, this looks like a phone cord. And they're like, phone cord? What's a phone cord? We just don't see those anymore. But, you know, I show them, I remind them, and they're okay. So, but that polymer actually starts to do this on its own, all because these atoms like to interact with each other. Now, I could spend a couple more hours actually talking to you about why that actually is the case, but they do. And so what is this goes on to do what you see here. It starts to take and it starts to compact and starts to, I'm going to use another term here. It starts to bond with itself, bind, however you want to define it. It starts to interact with itself. So you can imagine here, the more amino acids you actually put in this thing, the longer they get, the more complexity you add, the more complicated that interaction starts to become, okay? So that leads to, those sorts of interactions lead to what we see here. So this is a representation of a protein, and if you look real closely on here, it might not be very big on your screen here. This is a representation of a protein called myoglobin. This is real. This looks like artwork, but this is real by all the methods that we have to take and probe protein structure and understand where atoms are and amino acids are. This is extraordinarily real. And it forms for the reasons that I showed you on that previous slide, which are that you've got one atom that likes to be next to another atom. And so even though it's not shown here, there are some interactions that are happening here. There are interactions that are happening there. These things are coming together. They like to interact. This protein, for example, every protein, when a protein actually folds here, you actually get structure. And that structure ultimately leads to some sort of function. If you have a protein that basically is made, and proteins are made inside of cells, and that's a whole other couple hours I could chat with you about, but proteins are made inside of cells. If they stay like that, they have no structure. And typically, very typically, you're not going to have any function. So these interactions that result from these molecules basically coming together, coalescing, in doing that provide function. So myoglobin, some of you may know that this is a really important molecule, protein that carries oxygen. It resides in your muscle. Here, this actually is a picture of myoglobin that comes out of a whale. And then, as I said, this is real. We know that in the snapshot in time that these are where the atoms are. We can pick out where the atoms are in amino acids. We know this forms, and when this forms properly, in this particular instance, oxygen binds. Okay. So amino acids assemble into proteins. Proteins actually do this. It gives them function. Now, what you need to know about folding is that this happens, right? All of us are well acquainted with the fact that this happens. Now, it almost seems sometimes like there's no particular reason for this happening. But in essence, this happens because of the characteristics of the molecules that are in that substance that's there. Here, this phone cord, right? There's plastics, there's wires inside of that cord, and this just happens. Now, it seems sometimes like this happens just to infuriate you, right? We've all had those. Maybe we've actually bought that little gizmo that goes on the end there that's supposed to wind and help you prevent this sort of happening. But try as you might, this is going to happen. Okay, and the same thing is true with this. This is going to happen. Okay, and that is not because these molecules, atoms are smart. It's because that's what they do. All because one atom likes another, they get together, they hang out. I'm going to talk about why they actually like to get together and hang out and why they stay like that. And I'll talk about that in a moment. So I've given this slide away already here. The folding, the process by which this becomes that, okay, highly technical cartoons there. The process by which one thing turns into the other is because of those interactions, but more important than that, when you make those interactions, when you make those bonds, when something from over here finds that it likes to interact with something from over there, you release energy. You release energy. Now, that's the normal thing in this universe. It's a normal thing in this universe that things in this universe like to reside at a low energy state. Okay, so here I give you two examples, right? Okay, here are two of our fur balls here in the Patton household. And you actually see here one that has achieved a very low energy state, right? And one here that actually is not quite there yet. All right, so but what has to happen here? What has to happen here for cat number 3 here, and I'll classify her as cat number 3, in order to achieve a low energy state? Well, everybody's observed this. If you're a cat person, a dog person, whatever, right? You know, there are some kind of gyrations, manipulations that actually have to happen. And eventually, this one here is going to look like that one, at least for a certain amount of time, right? At least for a certain amount of time. Now, I want to draw here, which is probably the most technical diagram that you're actually going to see me draw in this talk here. And it's something that I feel is worthwhile understanding here. Let me draw this. So I'm going to start out here. I'm going to go up here, and I'm basically going to do that. All All right, so I'm going to say that this is start, finish, okay? And I include these kind of, normal everyday pictures here to kind of give us something to really hang our hat on. So I'm going to go start to finish, and this is energy. This is energy over here. So Cat walks in the room, right? Cat walks in the room. Cat has to go through some sort of gyrations here, whatever, find the right spot, okay? And so here, we're actually making a bond, making an association with the surroundings here in order to get just right. And so it takes a little bit of energy here. You've really got to make a little bit of investment of energy there to kind of get things quite right. Now that energy can be displayed just kind of as moving around, okay? That energy can be, so here we're sitting by baseboard, that energy can be given off by that baseboard there. That may be helping with that attraction there, but you get a little bit of moving around here and you get what is a very high energy state. Now you can do one of two things, okay? Again, you observe this happening in everyday life. So one of two things here, you get to the point where you're about ready to kind of go there, okay? Go here, which I'm calling finish. And you may decide, well, yeah, it's not quite time to do that, right? And so you're kind of back in this state. But you get to this point here and notice what happens. You have two choices. You can go back to there or you can go back to here. Now, if you think about this and what nature actually likes to do, if it's given a little bit of a nudge, it winds up going down here. And if you look at the difference in energy from where you started there to where you finished, what's happened is you've given off some energy to your surroundings. You've lost something. You've lost here what you're really doing here, where cat number one here, so I guess I call her cat number three. This is the one who's in charge. Cat number three here basically has kind of moved around and kind of gotten it all figured out and then goes, right? And there's a loss of energy. There's a loss of energy here. So this cat in this orientation, in this conformation here, is achieved a low energy state. Okay. Now, not unlike what actually really happens to, we want to look at our cartoon version of this, what really happens up here is that there has to be some movement, there has to be some twisting, there has to be some coming together, okay? But just like up here, when you have the twisting and the movement and the things come around in just the right way, it settles in and you get things associated in the proper way. So you wind up getting all of this, which is really random. And it's kind of like, I'm just going to settle in here. Boom. I've settled in. I'm stable. Okay. At least in their case, you get 20 minutes, maybe half an hour if you're lucky here in the case of in the case of these guys. But you come to that lower energy state actually, which is stable. And that's so important. Nature does this all the time, that things actually go to those low energy states, right? So this is one of the reasons that we sleep at night. We actually get to a low energy state. So I want to bring this back around to proteins. And I want to talk to you about this energy in terms of proteins here and bring up some examples that we're all familiar with, more or less familiar with. So one of the first proteins for which the structure was solved, matter of fact, the first protein where we actually understood the entire sequence of amino acids was insulin. And we all have some idea that insulin is important in sugar metabolism, helps get sugar inside of cells. Without it, it's not a good thing, right? You're very dire circumstances if you don't have your insulin system working properly. I note here, for example, this was solved by an individual who won the Nobel Prize for his findings in being able to do this. What's interesting about this individual here, and kind of a tie into the topic here, so Sanger wound up being a biochemist, person who investigates molecules that participate in the processes of life. He wound up coming around and doing something that is still, his methods are still important for what he did for proteins. But right here, every one of our COVID tests that happens by sequencing. So we have a couple number of COVID tests. We have a test that basically is an antibody test. That's quick, 5-minute test or 15-minute test rather. PCR test is kind of related to that. But if they really want to understand what strain of COVID you have, you actually sequence the nucleic acid that actually is found in that. And so his work here is still relevant today, because what he did actually with later on, after he got the protein stuff worked out, got into sequencing nucleic acids. That's still at the heart of how we determine nucleic acid sequences and ultimately do identification today. So won 2 Nobel Prizes for 2 things that were incredibly important. So what did he first of all do for us that's so important for us? The first thing that he basically did for us is helped us understand the sequence. So right back to my first slide here, understand the sequence of proteins, how those amino acids are linked together. If we know that, we know an awful lot about a protein. So this is small, this is simple. Mega importance as a protein in living systems, in mammals. But for us, we needed to understand, if we're going to learn anything about a protein, we need to be able to understand its sequence. Okay, he did that for us. He did that, and his researchers who worked with him helped us understand that. Later on, we developed a technology that I've already shown you here that allow us to understand how those amino acids are oriented in space. That ultimately gives us 3-dimensional structure. The 3-dimensional orientation of this stuff is everything, okay? Now, this comes together, even though it's small, this comes together because of the things that I've already shown you, like these things interacting with one another. It's really simple when it comes down to it. Basically, all this stuff comes together here, forms this sort of structure in space in solution because it's happening there. Why does it happen? It's not smart. It's just the way that things like to be attracted with one another. Okay, so that's insulin. It's small, it comes together, it has a really important function. Structure for us gives function. All right. So this was a molecule that I got introduced to during my postdoc work, and I spent a lot of years working on this and working with it in doing different investigations. We're kind of up a couple steps here on the ladder here. We have here, so this was 2 pieces. If you notice the color coding that I actually used here, these are 2 pieces that need to come together. 2 pieces, 2 peptides, 2 strings of amino acids come together here. This is 7, okay? And I've tried to color code this. Here's 1, here's 2, and it's a little bit hard on the eyes, but we actually see 5 in here if you can sort out those colors. All of this comes together, and you may or may not know what this actually do. This actually winds up being, this is nasty stuff. This can kill a person by giving them really bad diarrhea, drains them of water and electrolytes, and is deadly. We now have really basic and fundamental treatments for this that don't really involve anything with structure, protein structure here. You know, give them the right antibiotic. Cipro, for example, can be used to treat this disease because it kills the bacteria that actually cause this. But at the heart of this is a protein that's made by an organism that comes together because all these things just stick together just right. Again, not smart. It's just the way things like to come together. And what's really important about this, a lower energy state is achieved. All of these guys at one point, all 7 pieces were like this and free floating, and they turned into what you actually see down here, where basically these are almost like they're designed by an artist. They're artistically folded and associated and very stable. And this one actually has a number of nasty things it actually does, and it's all a result here of structure. If you get this thing to form a structure, which it seemingly knows how to do because the right things stick together, you're going to have some function here. Now, the more bonds, okay, the more stable the structure. So this thing, for example, is actually made by bacteria. It winds up in water supply. It can exist stably in water supply. It can make people very sick. It's nasty little stuff that sticks together just because it sticks together. Here's kind of another important molecule that has been some stuff that I've worked with here. This name is probably less familiar to people. It's ferritin. Ferris actually maybe gives some information to its function here. it starts out as simply enough, just one of these 242 amino acids. So that's kind of long, but that's not the longest thing that actually exists in nature. And so that actually comes together. It looks like this. You got multiple phone cords there that interact with each other, again, because it likes to stick together. It knows how to find, and it bumps in each other and compacts and fits together. What's interesting about this is like, okay, here's one chain of amino acids. What you may not know is that 24 of those chains come together and they form what I like to call here a molecular soccer ball. So this is kind of like a molecular soccer ball. And why do they actually do this? Well, we know the answer to that already. We know that things like to stick together, and they do so, they achieve a low energy state. Now, 24 things, bringing 24 things together on their own. Again, I'll go back to the cat analogy here. It must be like herding cats, and that comes from a Super Bowl commercial many years ago. But to get that to come together, again, it's almost like it's kind of smart, but it basically knows how to stick together. All right, we know when things actually stick together here that they achieve a low energy state. Now, one last example here that I'll bring up here is the protein beta amyloid, or any proteins that actually have amyloid-like properties that actually can wind up folding and forming things. So we have, you see scribbles here. I like the diagram because it looks like my scribbles that I make here. We have proteins that basically start out, they basically take and start out, they can stay kind of just all sorts of whatever, or they can become more highly organized. And I didn't use this term yet. So I believe I used a term, those things that look like phone cords, those things are called helices. There are some other things that we see in structures, and they've been up above, I just didn't point them out. They're called beta sheets. And what happens with proteins like this, the beta sheet is an extraordinarily stable structure, much more stable actually than helices. And there are reasons for that, energetic reasons for that, conformational reasons for that. But what happens in these particular proteins, they form this stable structure, and this stable structure here kind of seeds the structure around it, and it actually builds associations, and more and more and more come in. The most stable structure that forms from proteins that have amyloid-like characteristics or amyloid protein are the fibrils. So we actually see a term in here, are these fibrils, where you get things just lined up so neat and tidy, really, really tightly associated that they just start to stack and stack and stack and stack. It's incredibly stable, and it also takes and it seeds association with other proteins. That's a little interesting. So low energy state. Now I'll bring this back up here. So my diagram from up above here. So if you actually started out here and you actually want to get something that's folded, you're unfolded, here you're folded. If you actually want to take and you want to go back this way, right? If you want to go back up this way, your energy there and all the gymnastics and whatever you're going to do in order to get back up there, right? It was only just a little bit to get up to the top of that. Look at what you actually have to do if you actually want to get back to where you started from, if you want to unfold and break those associations. There's more energy into them. So this is why a low energy stable state is such an important thing to fully understand, because once you've actually lost that energy, to reverse that, and there's an awful lot of research going on, for example, how to reverse this, how to tear these things back apart. But really, it's actually going to take some energy in order to figure out, well, it's going to take the energy to figure it out, but it's going to take some energetics or some molecule in order to get in there and kind of break this up and tear this apart. It's not going to be an easy task to do, is basically to reverse that stuff, to reverse that stable of a protein structure formation. Now, let me go back to these guys again here. So one of the things, building off that commentary on amyloid proteins and the structures that they can actually form here, is that one structure here actually can influence the structure of its surroundings. Now, This actually works better when these structures are actually in more intimate contact with one another. And I will point out here, there was one picture I removed of a family member here where the family member actually took and decided to just after work, just take and sit down for a minute. Cat number one comes up, associates, and before you know it, both of them achieve a low energy state. Okay, but I took that picture out of that certain individual in my household in a low energy state with furball number one here in a low energy state. You guys get the idea of what happened there. But that's really, really important. That's really, really important, that the energy level of a structure and the fact that one interaction with another leads to an even lower energy state. So anytime you get two things, this may have some structure, this may have some structure, they associate, they lose more energy, they become more stable. And that's where I move into talking about the COVID spike protein. And hopefully I've done a good job in kind of setting up for you the importance of structure, stability, bonding, and energy here. So here's a version of the COVID spike protein that you perhaps haven't seen before. We as protein scientists have a ton of resources at our disposal, and it allow us to view some things here. So I'm going to go out to one of those resources right now, and I'm going to bring it up on your screen. Hopefully you should be able to see this. So this representation is of the COVID spike protein. So we can go back, and matter of fact, I'll do that right here. Let me go back here for a second. I take it for granted that we're all on the same page here with terminology, but let's actually go back here. I think everybody knows what this is, but let's just make sure. So up here, we've all seen this, right? So you see this constantly, maybe less so because we've seen it so much now. You see this structure constantly. We're looking at those guys right there. I'll turn my pen a different color here. We're looking at these guys right here. It turns out there's about 30 different proteins in this virus particle. And the one that actually is seemingly most important for function here is the one that helps this thing interact with a cell over there. Okay, so these are our spike proteins. That's what I'm going to be referring to here. So let me get myself back here and then go over to the other screen. So there we are, and I can come back to there in a moment. Let me go back out to my other view here. Boom. So here's a resource. This is the rcsb.org, the Rutgers Center for Structural Biology. It's the home of what we as protein chemists call the protein data bank. Another word for this is pdb.org, and they both kind of get you the same way. The tools that we have here, this looks like an artist rendering. This is a real vision for where the atoms are in this molecule. So what you see here, and I can't annotate this screen here, but hopefully you can see my mouse. Right down here, where you actually see this thing right at the bottom there, is where that would bind to the surface of the virus particle. It would actually be embedded in the surface of the virus particle. So everything that's up here is where this is actually going to interact with the cell. So we get an opportunity here. We can zoom in, we can look at this, we can understand the orientation of things. If you're really good with colors here, maybe you can see that there are 3 different colors. There are 3 strands of amino acids here, 3 chains. And as my screen showed here before I zipped out to this view, over 1,140 amino acids. Think of the complexity in this thing. So I'm going to bring up a view here. I'm going to bring up a view here that's going to show you, this kind of almost seems like an artist rendering of what's actually going on in this thing. There are various ways that I can view this. I can see elements of the structure. I can see where beta sheets occur. I can see that the core of this is actually stabilized by multiple helices that are actually interacting here to provide a really strong, but probably somewhat flexible core to this. And I want to take and show you here another view of this. So I'll show you the complexity of what really this thing is. So here, I just clicked on a view called space fill. And now this actually shows us the position. There are some atoms that aren't able to be solved in the structure for one reason or another, but it shows you the position of every atom in this thing. It gives us an idea of what this really looks like in space as this on the portion on the surface of a virus particle would approach a cell. So your cell would be over here. And now that basically are amino acids here that are seeking to interact favorably with other amino acids that are over here. Again, we know why they do this. Those interactions like hydrogen on nitrogen interacts with oxygen, it comes together here. There's good three-dimensional fit, boom, lower energy state. So what makes this protein important in the function of this virus is the fact that it has found something else in nature that it interacts with stably due to those sorts of interactions I've been discussing here. And it forms a stable bound product that's energetically stable and that will persist. That will persist for all the reasons, everything in the half an hour schooling you've got on protein structure and why it fits together. For all those reasons, this binding up here to the other cell occurs for all those reasons that we know. Now, so how do we use tools like this? How do we use tools like this? Well, one of the things that we actually can do, and there's complicated ways to do this, we can look for drugs. We can look for molecules that have particular structures that may fit someplace on the surface here and may actually inhibit. So here's this thing, and here's the surface here. Maybe something would come in. Maybe here's a drug that comes in and basically stops one of the things interacting with the other. Maybe there's a drug that we can put in here that maybe just doesn't allow. You can see, let me rotate this a particular way here. You can see that, and my computer's lagging behind here a little bit. I'll leave it like that. So if your cell's over here, and this wants to interact with the cell, some of this needs to move a little bit. We call that a conformational change. Some of that needs to move. It needs to be tweaked, kind of like those cats slowing down to their low energy state. I mean, that needs to just, some rotations and some molecular movement needs to occur there. Maybe we can find a drug that actually prevents that from happening. So this thing maybe still has the opportunity for some of it to bind, but you find a drug here that doesn't allow all of it to bind and all of it to interact. So this is the COVID spike protein. And as somebody who works with proteins, those are sorts of things that I consider as I look at proteins and consider what their function may be. So I want to talk about the virus. Now, I will preface what I'm going to share with you here by the fact that my expertise is basically at the molecular level. I'm not an immunologist. I understand portions of some of what happens here from an immunological standpoint. I understand portions of what happens here as the proteins are made and processed by cells. But I just want to talk about mutants and how they form and why they form. I think everybody here probably has a reasonable understanding that basically the virus particle here is going to come in. It's going to find something on the surface of your cell. You may know this of this thing called ACE2, angiotensin converting enzyme 2. It binds to that. There's some things that actually about that binding process here that trigger this thing to be taken inside the cell. And then once this thing is inside of a cell, there you have it. What's going to happen here? It's going to release its nucleic acid material. It's going to take, and that's going to be replicated. It's going to be reproduced. And then what happens is that not only are you actually taking and replicating that material, you're actually taking the genes that are in that material. The genes that are in this material also are going to code for proteins like the spike protein. So you're basically giving the cell the information to make multiple copies of itself. This virus brings with the information to just make multiple copies of itself. So long story short, and lots of things here that are beyond my ability to talk about intelligently, but I can tell you the fact that when the nucleic acid material, the RNA in there is replicated, the proteins are replicated, look what starts to happen. You start to make more virus particles. And so those virus particles are reassembled, they're exported, and now the cell that was infected now takes this thing that actually and now releases it, okay? I want to make sure that everybody's okay with understanding here. Everything that I've showed you here is not the result of anything that we would actually consider alive. Yes, the cells here that the virus interacts with, they're alive. They have alive in biological terms is it has the ability to take and actually process energy, generate energy, work with energy. This virus is not alive. Everything that it does is because of the interactions at the protein level that I've been talking with you about. Now, except for the nucleic acids here. Nucleic acids have similar sorts of interactions that they do. Like molecules find the appropriate partners and that sort of thing. But the fact of the matter is why do mutants form here? Because the virus has an opportunity to replicate. So this whole process here of get this inside of a cell, make more copies is extraordinarily fast, okay? And this is kind of like Lucy working at the candy factory here. You know, there are going to be some errors in that. You know, as things actually speed up here, you're going to actually get some mismatches. You're going to get errors. So as you actually take and replicate this nucleic acid material here, you're going to get some errors. Now, that means that anything that actually is exported here as new virus may contain errors in it, okay? Now, some of those errors, and I can get less sloppy if I go slow here, some of those errors actually are going to be things that just aren't good, right? That aren't going to be good for the virus. It's not going to make a virus particle here that is any more stable, or is not going to make a virus particle here that's going to promote more stable interactions with the cell. Okay, those aren't the errors that we're concerned about, and those aren't the errors that ultimately we see. The errors that actually are generated, that are the ones that persist, are the ones that actually then are more stable. They carry through. They are the ones actually that we're going to see winding up doing this sort of stuff. Okay, this was an image out of an article I ran into this week on NPR. And it really kind of, I think, shows us here what happens when you have mutants that develop all due to errors. So you get an infection, you replicate. The more this thing replicates, the more chance for errors, the more chance for something that is more stable. And so what do we have here, right? So we have here, right, the Delta was one of the ones that spread so quickly, and that had consequences. Now those errors that actually are introduced into the proteins in the virus particle can influence binding, cell binding, pathogenicity by some other mechanism. Probably many of these mechanisms aren't yet understood. But some of those errors, for example, I mean, when's the last time that we talked about some of these, or these variants, and a mutant basically is one change, but you get a cluster of changes, and then you get families of things that are like the same. So we actually call those variants. Okay, so these basically, for whatever reason, they either weren't strong enough, they weren't stable enough, they actually didn't wind up in great part persisting. Now, it doesn't mean that they're not still out there. They're probably still out there, at least at some level or another. When they actually take and they actually get taken up and they get replicated, all of these now have the chance to actually produce errors. So how did Omicron come about here? Well, Omicron, and there are lots of theories like why this all of a sudden popped up, but Omicron came about because there was an opportunity here to actually take and allow this thing to replicate, become full of errors, but those errors actually wound up being favorable for it. And so my best guess here with Omicron is that the errors here wound up actually being errors that actually took and made the interaction with the receptors on the surface of the cell more favorable, more stable. For everything that you now know about protein structure here is the fact that you make an error, all of a sudden, okay, you get to lower energy, you're going to have something that's more infective here. So something actually made this more infective, made it more transmissible, and probably more stable structure, or the spike protein and more stable interaction with the receptor there is actually, at least in part, probably responsible for the spread of Omicron, and why that actually just seemingly took over, here in other countries as the variant that was most persistent, and is seemingly the one that's out there nowadays. Now, anytime that you actually take and do that replication, you're exposed to the process, multiple fast processing of building these virus particles, and you basically get to the point where you actually have the possibility of getting more errors and more stable structures. So I wind up here with a question for us, and you see here, are there more mutants in our future? Anytime that you have the ability to copy something, you're actually going to be, you have the potential to actually have errors in it, and not only say, are there more mutants in our future? Not only are there, what are in our future? Now, you know, we have good ways to avoid these things. And as I look out at everybody there, I see the mask, right? So actually keep this stuff out of the possibility of getting replicated whatsoever. The other that certainly is vaccination. So if you're exposed to this, if you get this inside of you, if you get it into a place where you might encounter this, and there's a chance to get it to bind in here or down in your lungs or wherever, that if you're immunized here, you have a chance that maybe those antibodies are going to encounter these proteins and count on the surface of the virus particle, are going to destroy that virus particle before it can become infective to you. So we know how to avoid these, the development of mutants. We know ultimately how to avoid the development of structures that are more stable and will interact with things more tightly, and will wind up doing this, okay? And that ultimately is what makes this stuff be something that's going to be very difficult to counter unless we simply avoid exposure to it at all. Okay, so hopefully what I've done here this morning, hopefully that I've given you folks a foundational understanding here of what proteins are and how I view things in my world. You know, when I actually take a look at that protein structure there's some interesting and detailed things that I actually see in it, and possibilities for maybe what we could do from a pharmaceutical standpoint. But ultimately here we have some good tools. We know what they are that actually are going to help avoid the generation of structures which are more favorable. There's use a term here that I actually have above here, and it's basically a term, it's just another fancy term for energy. So it's called enthalpy. When you actually take and you're up at the top of that curve here and you go down the other side and you've lost energy here, that is a process we call energetically favorable, enthalpically favorable. That's going to happen because that's what Mother Nature does, is achieve a low energy state. So I think people understand maybe how to try and avoid us having to deal with that. So hopefully we do. Okay. So I'm out of time here. I'm going to leave a couple minutes for questions if there are any questions. So thank you for the opportunity here to get a little bit of insight into what goes on inside of my head on a daily basis. And thanks just for an opportunity to talk here this morning.
Yeah, question, Wally. What causes Delta to wane? I understand what you're saying about Delta going over to Omicron. I understand that. But then why does Delta die out? Why isn't Omicron in addition to what Delta already is? And one other question is, well, is it possible that Delta as a variant comes back again?
So from my perspective, and what this is, so this is actually an artist's conception of something that we call a phylogenetic tree. And what scientists actually do is they look at the sequence of the nucleic acids, they look at the gene level, and they kind of understand how these things are all related. So Delta here is, I think everybody knows, Delta here has been a really nasty version of this stuff. And so how does it go away? It goes away because the carriers of it die, and it goes away because it doesn't get transmitted. Certainly, it's my thought here that, you know, if we still have instances of this being transmitted, again, I'm not an immunologist, but I look at this from a structure stability standpoint. If we still have instances where this is transmitted, can this further mutate? I wholeheartedly believe that this has the ability to mutate. It actually is, there's something about it that it not only certainly likes to interact with cells really well, but then it also has other features buried in its other genetics of its other proteins that actually make it nasty. Whatever the mechanism of this disease actually truly is, that there are other mutations in there that make it nasty. So is that something that's totally going to die away? You know, we have an idea how to control that, but if that is actually allowed to circulate and still be an infectious agent, then that I predict also will mutate. Same thing with Omicron here. Now, for whatever reason here, Omicron in vaccinated individuals did not appear to be as bad for them. But I think there's data out there that says Omicron in non-vaccinated individuals still has the ability to be nasty for non-vaccinated or unvaccinated individuals. So it's all kind of in the molecular compositions and the genes of these virus particles and how they actually affect the organism. It could be here. It could be here that Omicron actually mutates into something that becomes nastier. And, you know, that certainly would be worrisome because clearly Omicron here, you know, likes to interact with cells, right? So easily picked up. That would be kind of something that's troublesome if that begins to mutate in its other proteins and its other genetics that makes that kind of more horrible for whatever it is that you're dealing with in here. You know, that there's some more stable structure there and creates some, you know, some other element to the disease, whether it's the inflammatory aspect of the disease somehow or something that just makes it nastier for your body to deal with.
Wally, this is Charlie. In part of your discussion, you talked about drugs and their action on the proteins, and it sounded like it was more the drug almost coating the protein to make it less effective at binding. The question is, vaccines act in a different mechanism, and I thought maybe I'd ask if you could outline that.
Well, again, I'll share that I'm not immunologist, but so anytime that you basically have, and I'll go back up to this, either of these. This, by the way, is I stumbled upon this yesterday as I was putting a talk together. It's New York Times site, which is just phenomenally useful. So I encourage you folks to Google New York Times COVID tracker, and there's tons of good, really good information in there. As antibodies bind to things, you have a cellular immune response, B cells, T cells, that actually will come and will take and will recognize things as foreign, or proteins that will come in response to the antibodies binding will recognize things as foreign. And then those cells will deal with that. You know, they have ways that they basically can take and begin to destroy what they recognize as foreign, either through a process called oxidation or proteolysis. And so there are a number of ways that when an antibody sees something as foreign, it actually basically takes and can destroy it. So an immunization is good because you then generate antibodies to things that you haven't yet seen. So now you have those antibodies around, and you actually take and encounter this in the form of the virus instead of actually was just in that vaccine that helped stimulate the antibodies. Now the antibodies encounter the virus, and now you have this big response that destroys that. So from a drug standpoint, there are multiple different ways that you actually can take and you can envision a drug actually functioning here. And I liked your analogy here of kind of coating a molecule here. And so you can imagine that you maybe, if you have some drug here, and the scale here is like way out of whack. I'll tell you that right now. So from a molecular standpoint here, your drug is probably about yay big compared to this overall molecule here, but I'll draw it bigger here. So let's just make a drug that likes to interact with this three-dimensionally. So like the three-dimensional interaction is like this, right? That basically fits together, or is like that. That fits together. It isn't necessarily like this, you know, that isn't a good fit. That's a better fit. So you make some drug there that maybe binds, interacts, and maybe does it stronger. So up here is a receptor on the surface of the cell, and maybe like right there is where this portion of this likes to bind. So you develop a drug that binds really well here, so it can't bind. So that's one possibility of how a drug maybe actually could inhibit this. inhibit the binding and lead to some sort of therapeutic approach to this thing. And another one, as I said here, it's very clear. We know that there's very clear here. We see here, there's a whole bunch of space here. There are gaps that are in there. It's hard to see when I turn on that space filling model with all the atoms in it. But if you actually could take something here that basically just wedges itself in there and is really stably bound, then if this needs to swing up here in space in order to do the interaction there, if you prevent that, it's a fancy term that we use for that, it's a change in conformation. So we say, we hand wave this away and say, oh, we can prevent a conformational change. If we can basically tuck something in here, wedge something in there that doesn't allow that tweak, that change to occur, then maybe that can actually inhibit the interaction with the cell, because the protein's not quite in its coziest, most stable conformation in order to get that interaction to happen. So no doubt that there are people out there who actually are all over this protein. We have ways with computers to basically take any molecule that you can imagine that has ever been made and synthesized and just kind of like run it over the surface of a protein here and kind of figure out where it fits better. Charlie, I know you're a big puzzle person. And so you kind of get those difficult pieces sometimes. You're not really sure where they go. You kind of just take and kind of rotate them around and kind of move them around the whole surface of the thing. At least that's what I see in my family. And we can do that from a computer standpoint. We actually can take molecules and we can just push them around the surface of this thing and kind of see where they fit snugly and have those favorable interactions. And so I'm sure that there are scientists out there running computers day and night to figure out what molecules they actually have on hand that fit in different places, or what's even better than that, actually taking and generating molecules that say, okay, I know what this actually, I think it looks like in space and how it's oriented. You know, I need like a hand here with like 10 fingers, but I can build that drug in order to fit in there. Let's make it and put it in there and stop that from happening. And let's see what happens to generate a therapeutic. Long answer to a short question.
So maybe a related question would be, Moderna is supposed to work differently than Johnson & Johnson. Can you speak to that?
Not in great detail. Interestingly enough, before I actually came to LVC, I was looking at taking all this protein technology and working in industry for a vaccine company and the technology has in those 20 plus years, that technology has become just new and very interesting and whatever. My understanding of how those two things differ is with J&J, there's a more traditional technology of producing things that generate antibody responses that are basically based on the cold virus, the adenovirus, and inserting sequences in proteins that belong to that, and then allowing your body to generate an immune response towards that. And what the Moderna version of this is totally different from the standpoint of they give you nucleic acid and your body generates a short segment of the protein. Then that protein actually gives you an immune response. So they're different. They're different in how they actually take and they deliver the material that actually takes and allows your body to generate an immune response. Now, as I said previously, not being an immunologist, that's kind of my rough understanding, especially of how J&J works. But both vaccines present your body with a short piece of protein that allows your body to say, okay, something here is foreign. So let's say, for example, and I don't know where the sequences are that they actually made in either vaccine to actually take and allow your body to see it as foreign. But let's just say they maybe take a 10, 20 amino acid stretch from like right there, because this is where this binds the cell. Maybe that's what they actually did and say, okay, well, you know what, I'm going to put this in a vaccine. The nucleic acid genetic code that codes for that little piece of protein. So that little piece of protein there, not infectious in any way, shape, or form, but if your body generates antibodies against that, maybe an antibody comes in here. So there's an antibody coming in here and recognizes that and starts to degrade that. So the virus actually no longer has this portion on it that can't be infected or can't be infectious to you. So So any vaccine is going to present your body through some mechanism or another with a short little stubby piece of something that isn't enough to make you sick, but is an important portion, what we call as an antigen, right? So we actually take and present you with the antigen, so your body produces the antibodies against it. And so now your immune system kicks in and sees that, as recognized that as foreign, and tries to destroy it. So they're the differences, and if you will, the similarities for how those approaches are occurring in those vaccines.
Thank you. If there's not any other questions, it is a little past 10 o'clock. And we thank you, Wally. That was a great presentation. I feel like I got a year's worth of education in an hour. So thank you very much for presenting this morning.
Again, thanks for allowing me to be here, to be with all of you, and you get me talking, and this is what happens. So I hope there was something good here for everybody today. Thanks again.